hope that you will get as much out of this as I have in putting it together and implementing it over the past few years. And with that, let's move forward. All right, so what we're going to do today is go through a number of things. We want to know first and foremost what's driving this whole development of QTLs, and then to define them. And that's an interesting conundrum when we talk about parameters and so forth. So you'll see the clear difference in a few slides to come. We're going to look at part of a protocol to identify how to identify critical processes that could impact subject safety or data integrity, how to document that development process, how to implement your QTLs that you've established, how to track and assess the data to establish if what you're looking at is within your quality tolerance limits, and then responding when those QTLs are exceeded. And also understand that QTLs evolve over the life of a clinical trial. And then possibly how to modify those QTLs depending on the circumstances of your clinical trial. And then documenting any changes to those QTLs because those changes must be justified. Okay, so I'm sure everyone has seen this before. Risk-based quality is a cycle. It's never, ever static. So, and we don't take decisions or implementation of solutions in a vacuum because things continue to go on around them. So those decisions are continually reevaluated to assure that they're producing a desired outcome. Okay, so what promulgated development of QTLs? Well, the EMA, in their clinical trials regulation, Article 47, made ICH GCP legally binding. So that shifted the judgment of the sponsor to legal obligation, right? So the part that addresses this in ICH E6 R2 is 504, that section on risk control. And in essence, it says to determine the point where, if met, the ability to draw conclusions from data is lost. So this is broad. So I want you to think of quality tolerance limits as broad, but also could be narrow. So you really need to look at the spectrum here. So some of the questions you might ask yourself in your ability or inability to draw conclusions from data is, when will power be lost? Power of the test. When is data integrity lost? So those are the pretty broad concepts and encompass many moving parts. In continuing the requirements that propelled the development of quality tolerance limits, R2 Section 507 speaks to risk reporting, and it goes further to say that the sponsor should describe their quality management approach that they're using in that trial and summarize important, and that keyword is important, deviations from the predefined quality tolerance limits and what they did about it. And that needs to be reported in the clinical study report. So let's define them and understand what they are. So according to Deming, tolerance limits are acceptable defects rather than zero defects. We're all human. We make mistakes. Which are acceptable, which are not. Carry that thinking over to a clinical trial, right? In that context, we look at random errors versus systematic errors, and these are statistical terms. So in random errors, you'll see the ovals on the right, there's no identifiable common root cause, and when you take their impact as a whole, they usually don't change the emerging data pattern in one direction, right? They're scattered, they're random.